Hi everyone, and welcome to today's session where we're going to look at carbohydrates in our series on biomolecules. And by the end of this session, we expect to be able to classify carbohydrates into various groups, describe the structure, chemical and physical properties of carbohydrates as well as their biological functions, and also be able to outline disorders associated with carbohydrates. So to start off, let's look at an overview about carbohydrates. And carbohydrates are defined as aldehyde or ketone derivatives of the higher polyhydric alcohols. And they're the most abundant macromolecules in nature. However, in mammalian organisms, carbohydrates constitutes only about 1%. All carbohydrates contain carbon, hydrogen, and oxygen. Many but not all carbohydrates have the empirical or general molecular formula of CnH2nOn. And of course, C stands for carbon, H for hydrogen, and O for oxygen. And the value of N is equal or greater than 3. However, there are some carbohydrate compounds that may have nitrogen, phosphorus, or even sulfur. When it comes to classification, carbohydrates can be classified into 3 major groups. We have monosaccharides, including glucose, and this consists of a single polyhydroxyl aldose or ketose unit. Oligosaccharides, on the other hand, consist of short chains, monosaccharides, joined by glycosidic bonds. And here we're talking about 2 to 20 monosaccharides units. Sucrose is an example of oligosaccharide. In regards to polysaccharides, these are sugar polymers which contain more than 20 monosaccharide units, and starch, which is found in plants, is a good example of polysaccharides. So let's look at each group in detail. We start off with monosaccharides, and they're also known as simple sugars. They cannot be hydrolyzed further into smaller molecules. Looking at the properties of monosaccharides, these compounds are white solid, they're water soluble, that means they are not soluble in nonpolar solvents and often have a sweet taste. The formula for carbohydrate is CH2O into brackets N, where the value of N is between 3 and 7. And so classification according to the number of carbons, which range from 3 to 7. If a compound has 3 carbons, it's called a triose. Glyceraldehyde is an example of a triose. In case of 4 carbon is a tetrose, whereas 5 carbon, these are pentoses, 6 hexose, including glucose, and also the 7 carbon heptose and pseudoheptulose is a good example. It is important to note that most of these monosaccharides are energy-giving compounds, including glucose. Some, including ribose, are involved in formation of nucleotides, glyceraldehyde, erythrocytes, erythros, and also pseudoheptulose. We find them as intermediates in various biological activities in the human body. Monosaccharides can also be classified as according to the carbonyl group, and here we're talking about aldose and ketose. Looking at aldoses, these are monosaccharides whose carbon skeleton carries an aldehyde group at the end of carbon chain. And an aldehyde group, this is a carbon atom which is double bonded to an oxygen and at least 1 hydrogen atom. And so looking at these examples that we have here, we have glyceraldehyde, and you can see at the end carbon chain, we have the aldehyde group. So glyceraldehyde is the simplest aldose, and other examples of aldoses include ribose, glucose, and galactose. And you see that ribose is one of the components of DNA and RNA that is in the nucleotide, whereas glucose and galactose are majorly involved in energy production in the human body. Looking at ketose, these are monosaccharides whose carbon skeleton has a ketone group. And when you're talking about a ketone group, we're talking about a carbonyl group. And in most of these monosaccharides, we'll find that the ketone group is located at the second carbon atom. And looking at this simplest example of dihydroxyacetone, we can see that the carbonyl group is at the second carbon. The same goes for ribulose and also fructose, which are examples of ketosis. So looking at the structural aspect of monosaccharide, and here we're talking about stereoisomerism, which is a very important feature of monosaccharide, and the presence of asymmetric carbon. And this is a carbon atom to which 4 different groups or atoms are attached. And so the presence of asymmetric carbon in a monosaccharide gives rise to the formation of isomers of that particular compound. So such compounds will have the same composition, but only will differ in the spatial configuration. And so we can say stereoisomers are compounds that have the same structural formula, but differ in their spatial configuration. When you talk about spatial configuration, this is a fixed 3-dimensional arrangement of a particular compound atom in space. Now, it is crucial because it determines how a molecule behaves, how it interacts with other molecules in biological properties. We will look at glyceraldehyde as a reference carbohydrate. And glyceraldehyde, as we had earlier stated, it is a triose, and it's the simplest monosaccharide with one asymmetric carbon atom. And it exists as a 2 stereoisomers, and therefore it has been chosen as a reference carbohydrate to represent the structure of all other monosaccharides. And the first structural aspect is the enantiomers. And these are specific type of optical isomer. And the structures of the compounds form mirror images of each other. And these 2 members are designated as D and L sugar. Looking at glyceraldehyde, which we said is a reference monosaccharide for all other monosaccharides, if the OH in the asymmetric carbon is on the right, the sugar is of D series, and so this compound is D-glyceraldehyde. On the other hand, if the hydroxy in the asymmetric carbon is on the left side, the compound belongs to the L series, and therefore it's known as the L-glyceraldehyde. So this is what you said, that D and L-glyceraldehyde are used as parent compound to designate all other sugars or other compounds as D or L forms. And this strategy in other monosaccharides which have more than 3 is determined based, more than 3 carbon is determined based on the chiral carbon, which is farthest from the carbonyl group. And this carbon is also known as the penultimate carbon. It is also asymmetric because it is linked to 4 different atoms or group. So looking at, in case of glucose, as an example, in glucose, the penultimate carbon is carbon number 5. First, it is asymmetric, and it's also the farthest from the carbonyl group in regards to its asymmetric character. So the configuration of the hydroxy group using glyceraldehyde is on the right. If it is on the right, it will give rise to D-glucose. And if the configuration of the hydroxy group is on the left, it will give rise to L-glucose. And so 2 mirror images are produced. What is important to note is that the D sugars or D-glucose are the most natural, or they are the ones which are natural occurring, and the body is able to metabolize them because it has enzymes which are specific for the D-glucose configuration. Epimers, these are 2 monosaccharides which differ from each other in their configuration around a single specific compound or carbon other than the anomeric carbon. So we said that the molecules or the 2 monosaccharides are epimers to each other. Taking an example of glucose and galactose, these are epimers with regard to carbon number 4. So if we look at carbon number 4, glucose and D-galactose, they differ in the arrangement of the hydroxy group at carbon number 4. So in D-glucose, we see that the hydroxy group is on the right side, whereas for the D-galactose, it's on the left. Glucose and mannose are also epimers with regard to carbon number 2. If we look at the carbon number 2 of mannose, we see that the configuration of the hydroxy group at carbon number 2 is on the left, whereas that in glucose is on the right. The interconversion of epimers is known as epimerization and requires epimerases as enzyme that catalyzes that epimerization reaction. Ribose and cellulose are also epimers, and here we see that they differ in the configuration of the hydroxy group at carbon number 3. Functional isomers, here we see that the compounds have the same molecular formula, but they differ in their functional groups. For instance, glucose and fructose are functional isomers. They have the same molecular formula, but when you look at glucose, it contains an aldehyde group, whereas fructose has a ketone or keto group as a functional group, and so they are functional isomers. Anomers, this is another form of stereoisomers. Here we see that glucose can, in the cyclic forms, can either be alpha or can be beta, and the two molecules, that is alpha glucose and beta glucose, they differ from each other in configuration around carbon number 1, which is known as the anomeric carbon. So looking at the alpha anomer, we see that the hydroxy group is on the opposite side of the carbon number 6, that is a CH2OH. Whereas the beta D-glucose, we see that the hydroxy group is on the same side of CH2OH, that is carbon number 6, in the sugar of the sugar ring. And so we have alpha D-glucose and beta D-glucose, which are anomers, and this is in regard to the configuration of the hydroxy group at carbon number 1 of the cyclic forms of D-glucose, which is the reference monosaccharide we're looking at. And the same will apply for fructose, the same will apply for galactose in relation to their cyclic forms. So let's look at the features and also the Haworth projection. And when you're talking about the features projection in regards to monosaccharide, more so glucose, fructose, this is the linear or open chain structures of carbohydrates. On the other hand, the Haworth projection is a cyclic or ring presentation, which is the major way the monosaccharides are presented. And this is more of the carbons which have 5 and 6 carbons. So here we have a representation of glucose in the features projection. Remember, we have said it's linear, it's open chain, and a Haworth projection is when it is cyclic in nature. One thing we need to remember, Haworth projection will give rise to anomerism, which we've looked at. And this is what you say, that when you are talking about alpha anomer, the OH group or the hydroxy group on the anomeric carbon is opposite to carbon number 6, that is CH2OH. And for beta anomer, the hydroxy group and the carbon number 6, that is CH2OH, are on the same side. And the conversion of these anomers from alpha to beta or from beta to alpha is known as mutual rotation. So let's also look at some of the biologically important sugar, which are glucose derivatives. Upon oxidation, they lead to formation of aldonic acids, and oxidation of glucose in this aspect results to the formation of gluconic, uronic, which we find that uronic are components of mucopolysaccharides, and it's also required for detoxification. Reduction of aldose and ketone groups will result to sugar alcohols. This include glycerol and also inositol, which are intermediates of metabolic reactions. Amino sugars, now these are sugars in which the amino group is substituted for a hydroxy group, for example, glucosamine, which are components of mucopolysaccharides. Glycosides, they may be of two. We have two types of glycosides. We have the O-glycosides, and here we have hydroxy group on the anomeric carbon reacting with an alcohol. And this will result to the formation of O-glycoside. As we shall see, O-glycosidic linkage is present in disaccharides and also polysaccharides. The other form of glycoside is the N-glycosides, and this is formed when hydroxy group on the anomeric carbon reacts with an amine. And N-glycosidic linkage is present in nucleotides, that is RNA and DNA. So let's move on to the next group of monosaccharide of carbohydrates, and that is oligosaccharides. And as we had earlier mentioned, they consist of 2 to 10 monosaccharide units, and they are joined together by glycosidic or glycoside bonds. And this joining together of two monosaccharides takes place through a process known as condensation, because it results in the removal of water. And the most important oligosaccharides in nature are the disaccharides, since they are shown to provide energy to the human body. And the three major disaccharides are maltose, lactose, and also sucrose. So let's look at each disaccharide in detail, and we start off with maltose, and it contains 2 glucose units. So here we see that the anomeric carbon of the first glucose molecule will form a glycosidic linkage with the second carbon or of the second glucose, or with the fourth carbon of the second glucose. And so the glycosidic linkage of maltose that will be formed will be alpha-1 for glycosidic bond. So this is what you say. Carbon, the first alpha-D glucose will have the first carbon joining or forming a linkage with the carbon at position 4 of the glucose molecule. And so it will result to an alpha-1 for glycosidic bond. And this reaction, it's a condensation reaction resulting to the release of water. Now, it's also important to remember on how to count carbons in a glucose molecule. So we start at carbon number 1, 2, 3, 4, 5, 6. And that's why we're saying carbon number 1 and carbon number 4 of the second glucose are involved in a glycosidic linkage. And as a result, we see that maltose is a reducing sugar because the anomeric carbon of the second glucose is free, because it's not being involved in the formation of the glycosidic linkage. And so it can form an aldehyde group in solution. And it is said to be a reducing sugar because the free aldehyde group can reduce Benedict's and other reagents. So sources of maltose, majorly in germinate cereals, and it can also be formed during the hydrolysis of starch. Looking at lactose, now lactose contains 1 glucose and 1 galactose molecule. And here we see that the anomeric carbon atom of galactose and carbon atom 4 of glucose are involved in the glycosidic linkage. and therefore it is symbolized as beta 1,4 linkage. And therefore, lactose is a reducing sugar because we see that the anomeric carbon of glucose is free to form an aldehyde in solution. And so this is the linkage we're talking about, beta 1,4 glycosidic linkage. Why beta? Because galactose is the one which is giving us the first carbon. So sources of lactose, milk, which is synthesized in the mammalian gland. Sucralose is the other disaccharide, which is made from a D-glucose or an alpha-D-glucose and beta-D-fructose. And here we see that the anomeric carbon of glucose and anomeric carbon of fructose are both involved in glycosidic linkage. And therefore, we're talking about carbon number 1 of glucose and carbon number 2 of fructose. And so the linkage will be designated as alpha 1 beta 2 glycosidic linkage. Is it a reducing or non-reducing sugar? It is a non-reducing sugar because both of the anomeric carbon or the functional groups of glucose and fructose are involved in the glycosidic linkage, and therefore there is no free anomeric carbon which has a free aldehyde or ketone body. So sources of sucrose, fruits, honey, sugarcane, and also remember this is a source of energy. So other disaccharides are also there. We have the isomaltose, which contains 2 glucose, and the glycosidic linkage is alpha 1, 6. We also have cellulose, which contains 2 glucose units, but they are joined in beta 1,4 linkage. So looking at polysaccharide, which is the polymer or sugar polymers containing more than 20 monosaccharides units, they are the most carbohydrates found in nature, and they form high polymers, or the monosaccharides form high polymer molecule known as the polysaccharides. That's what you have said. So there are of 2 types. We have homopolysaccharides. When you talk about homopolysaccharides, we're talking about monosaccharides are the building blocks of these polysaccharides, but it's only one type of monosaccharide. When you're talking about heteropolysaccharides, these compounds or these polymers contain 2 or more different kinds of monosaccharides as a building blocks. And you're going to look at each in detail. So we start off with starch with an example of homopolysaccharide. And it is a polymer consisting of D-glucose units. It is insoluble in water because of its high molecular weight. And starch is a storage compound in plants. And it can either be amylose or amylopectin. Amylose constitutes of about 10 to 30% of the starch, whereas the amylopectin is 70 to 90%. So when you talk about the amylose, this is just the chain, the straight chain with no branching of the starch. And you have only one linkage, that is 1,4 glycosidic linkage. But when you're talking about the amylopectin, this is a glucose polymer which has 2 linkages. First, we have the alpha 1 linkage, which is made up of a straight chain, but then when it branches, it constitutes the alpha 1 to 6 linkage. So you can say the amylose is a branched form of starch. Glycogen, this is a storage polysaccharide in animals. Remember, we have said that starch is a storage form of glucose in plants, and then glycogen is a storage form of glucose in animals. And we see that mammals or animals prefer glycogen over glucose as a storage form of glucose in animals. And we find that the liver constitutes of about 10% of glycogen, whereas the muscle mass has about 1 to 2%. Now, glycogen is somehow equivalent to the amylopectin found in starch because it's also branched, but here we see that glycogen is highly or more highly branched compared to starch. Cellulose, this is one of the most abundant natural biopolymers. It's an example of homopolysaccharides, and because it consists of glucose monomers, which are linked through beta-1,4 glycosidic linkage. So the cell wall of plants are mostly made up of cellulose, and therefore they provide structural support to the cell. So it is important to note that glycogen, starch, and cellulose have glucose as the monomers. It is important to be able to know the differences between the linkage found in glycogen, starch, and cellulose. We've seen that starch and glycogen, they have alpha-1 glycosidic linkage. However, when it comes to the cellulose, we see that their linkage is beta-1,4 glycosidic linkage. Dextran, another example of polysaccharide. Here we see that the monomers are glucose units. However, they are linked by alpha-1,4 glycosidic bonds. This is a linear part. And the branching part, it has alpha-1,3 glycosidic linkage. And this polysaccharide is present in bacteria. So it has some medical importances. We see that the dental plaque is due to the dextran synthesized by sucrose by oral bacteria. Chitin, it's also a homopolysaccharide, which is composed of N-acetyl glucosamine as a monomers. And the glycosidic linkage found between the N-acetyl glucosamine is beta-1,4 linkage. We see that chitin is very important for structure as a structural polysaccharide found in invertebrates, including crabs, lobster, and also insects. Now, looking at heteropolysaccharides, now we said that they contain two or more different kinds of monosaccharide building blocks. Examples include the glycosaminoglycans, that is GAGs, and this consists of repeating disaccharides units. So they're highly negatively charged and therefore attract waters, contributing to the extracellular matrix viscosity and also resilience. Now let's look at examples of GAGs. We have the hyaluronic acid, which is composed of repeating units of glucuronic acid and N-acetyl glucosamine. We find that hyaluronic acid is abundant in connective tissues, in synovial fluid, and also the vitreous humor. So most of the functions of hyaluronic acid include lubrication, hydration, also they provide structural support. Heparin, which is fractionated from heparin sulfate, it's mainly stored in the mast cell. This is a type of leukocyte, and it acts as anticoagulant by enhancing antithrombin ring activity and therefore prevent blood clotting formation. And so it's important during blood transfusion. Damatine sulfate, it's composed of repeating units of hyaluronic acid and also N-acetyl galactosamine, found in skin, blood vessels, and therefore it contributes to elasticity and also tissue repair. Chondroitin sulfate, as a GAG, it consists of repeating disaccharides of glucuronic acid and also N-acetyl galactosamine. Keratin sulfate is also another form of GAGs, and it's majorly found in the cornea and also the cartilage. They provide hydration and also maintain tissue transparency and resilience. Glycoprotein is another form of heteropolysaccharides, and as the name suggests, it contains proteins which covalently link to short oligosaccharide chains, and they are majorly involved in cell recognition, signaling, and also have structural roles. And unlike the GAGs, glycoproteins, the carbohydrate portion is usually shorter and also branched. So let's look at some or mention some disorders associated with carbohydrates. We have galactosemia. This is a rare genetic disorder, and the body is unable to break down galactose, which is a monosaccharide found in milk. And so it is caused by deficiency in one of the enzymes involved in galactose metabolism. And if untreated, it can result to liver damage, kidney failure, and also neurological impairment. Fructose intolerance or malabsorption. We see that some individuals may have difficulty digesting and also absorbing fructose, and this will result to gastrointestinal symptoms, including bloating. And this may be due to deficiency of some enzymes which are responsible for fructose metabolism. Lactose intolerance, here is a condition where the body is unable to digest lactose, and this is due to lack of an enzyme lactase, which is responsible for breaking down lactose into simpler sugars, that is glucose and galactose, and therefore cannot be absorbed in the bloodstream. So having looked at that, let's finally look at some of the lab tests which can be done for carbohydrates, to test for carbohydrates in samples. And the first test is called the Molich test, which is a general test that is used to determine if any carbohydrate is present in a sample. So in this reaction, we have the concentrated sulfuric acid dehydrating the glycosidic bonds, and this will result to a purple or violet ring at the junction of the two liquids if carbohydrate is present in the sample. However, if there's no positive result, if there's no purple or violet ring at the junction of the two liquids, the color will remain, that is a brown greenish ring, and this shows absence of carbohydrates. Looking at the Benedict test, it is used to identify reducing sugars. And in this reaction, the reducing sugar contains a free aldehyde or ketone groups that reduces the blue copper 2 sulfate to a reddish or brick red precipitate when heated. And in case or in presence of a reducing sugar, green will show traces amount of the reducing sugar, whereas yellow or orange will show moderate. Brick red precipitate will confirm high concentration of the reducing sugars. However, a negative result is when the color just remains blue, just showing no reducing sugar. So having known this, you can go back and look at which monosaccharides and which disaccharides will tend to show or will test positive for the Benedict test. Looking at the iodine test, it's used specifically to detect starch. Remember we said starch is the storage form of the glucose storage form in plants. So we see here iodine slips into the helical structure of amylose in this reaction, forming a complex that absorbs light differently. So in case of presence of starch, it gives a positive result, and it's indicated by a blue-black color, whereas the negative result, the solution remains yellow or brown. That is the color of iodine. But then if you heat the blue-black solution, the color tends to disappear because the structural or the starch helix unravels, and then it reappears upon cooling. Buffert's tests, this test distinguish between reducing monosaccharides and also reducing disaccharides. And this is based on the speed of the reaction. So it's somehow similar to the Benedict, but performed in a weakly acidic medium. Monosaccharides, of course, are stronger reducing agents and therefore react faster compared to the disaccharide. So expected results, monosaccharides will form a brick red precipitate within 2 to 3 minutes of boiling, whereas the disaccharide precipitate will form only after prolonged boiling, that is about 10 minutes. In case of absence of carbohydrates or reducing monosaccharides and disaccharides, the solution remains blue. And finally, the Seliwanov test. Now, this one is used to distinguish between sugars with ketone group including fructose, and those with an aldehyde group, as we had mentioned earlier, that is majorly glucose. So in this reaction, ketoses are dehydrated more rapidly than aldehydes. That means if it was between glucose and glucose and fructose, we see that fructose will be dehydrated more rapidly compared to glucose, but the hydrochloric acid, and therefore we react and form a cherry red colored complex. And for aldose, it will form a faint pink color, which tends to develop more slowly compared to the ketose. So it's able to distinguish whether a sugar is a ketone or an aldehyde.